And we'll admit everybody. Hello and good afternoon, everybody, and welcome to the last of these STFC Food Network webinars, a special season of webinars to mark World Food Day. We've had 4 great webinars in the week, and I'm sure this last one will be just as good as all the rest that we've had before. There's been some terrific discussion after each of the webinars, lots of great ideas being shared. And this afternoon, we're going to hear from 3 researchers working in the wide-ranging field of ensuring food safety. Their research is an example of the multiple challenges around ensuring food is safe for human consumption and quality is ensured along the supply chains. So I'm now going to hand over to our chair for this afternoon, who is Sonal Chowdhury.
Thanks, Eric. And hello, everybody. I am Professor Sonal Chowdhury. I'm a PI, the lead for STFC Food Network Plus. And I'm really pleased to see that, these 3 speakers will be presenting different aspects and different projects on the food safety. So I would like to welcome our first speaker for this webinar, Professor Nicola Holden. She is a professor of food safety and Scotland's Rural College. She'll be presenting on taking a big data route to identify notorious foodborne pathogen. And I'm daring not to pronounce the pathogen's name here, but probably Nicola, you would be best to present and let us know more about this project. Over to you, Nicola.
Thank you very much, Sonal, and thanks everyone for letting me share our findings from our nice little project. So I'm going to share the screen now. Please let me know if you can't hear me or if something is unclear. But is that okay?
Yeah.
Cool, thank you. Okay, so I'm afraid I'm going to be looking between 2 screens, so I'm sorry if I look like I'm ignoring you. So what we're talking about today is the sugar toxigenic E. coli. And so this is a nice project looking at how we potentially can detect the pathogen. And this was done in collaboration with Martin Wynne at FTSC, and he's based at the Harwell Centre. And we had another food safety expert called Tim Dolman, who used to be with Public Health England, but he's since moved to University of Utrecht. Oops. Next. That's it. Okay, so this is our problem, first of all, just to outline what we've got. So sugar toxigenic E. coli is a pathogen. I'm just going to call it STEC today. And this is a foodborne pathogen. And it's quite a notorious pathogen, probably best known as the burger bug, because it's been associated with undercooked burgers. And it causes gastroenteritis and hemolytic uremic syndrome, or kidney failure, and sometimes meningitis, and it can be fatal, especially in young people. So although it was called the burger bug, it's not just associated with undercooked meat, it's also associated especially with ready-to-eat vegetables and unpasteurized milk or cheese. So what I need to tell you what makes STEC a pathogen. So first of all, the toxin itself, and we've got different ways of calling the toxin, STX. But then also this bit, the O157H7 that you may be familiar with. So that's the O antigen, which is on the surface of the bacteria, and the H antigen is its flagella. Now, there's not just a 157H7, there's lots of different O's and there's lots of different H's. And then you've got the virulence factors on top of that enable the bacteria to colonize its host. So you need a combination of these different things in order to make STEC a pathogen. Okay, so what is the challenge? So O157H7 is our most common, or used to be our most common STEC. But actually, if you look on the graph on the left-hand side, the cases have been decreasing over recent time. And what we see on the right-hand side is the number of all the other STEC pathogens have actually been increasing. So they just get lumped together in a category called non-0157. The nomenclature is a blooming nightmare, but I hope you can bear with me. But the point is, what we've got is an increase in these other types of STEC. Now, just going further into the challenge, we have that increase, as I say, and there's calls for evidence from the World Health Organization and the FAO to see what's actually happening. We have, in the UK, we have pathogen surveillance programs, such as PathSafe, which is just initiated this year by Food Standards Agency. We need to understand the transmission rates of these bacteria. Because there's so many different O types, there's a big burden for screening in the clinical labs, and we need much better detection. So for example, a typical call might be a desperate plea for rapid or simplified testing of the non-0157 STECs. We can link up with technological innovations. Everybody knows about lateral flows now, so that's a perfect example of how we can use this sort of really nice little quick and easy devices to look for these pathogens. But there's a lack of selectivity. So when we look at just a presence of the toxin gene, for example, or one of the virulence factors, that's not enough. And what I've got on the right-hand side of this image, or this slide, sorry, is a risk matrix. And this is actually very helpful. So what it does is it categorizes all the different variants of toxin. Everyone's quite familiar now with variants of the spike protein, for example, for the SARS-CoV-2 virus. And just in the same way, you get variants of the sugar toxin. Some are more pathogenic than others. So if you combine that variation with variation in other factors, such as the virulence factors, we can map all of these together to make a risk matrix. So that works quite well, but it's still at quite a crude level. So we need to do something much more refined. And what we wanted to do was take a genomic-based approach. So working on the genomes of these bacteria. We have the advantage that they're bacteria, so they have much smaller genomes than humans or plants, for example. And what we have at our disposal are genomes from the Scottish Reference Laboratory, for example, or from other agencies. We have some good published papers on the prevalence and epidemiology. They're starting to come through now for the non-0157 STECs. And we have surveys. So for example, there was a survey of minced beef very recently, and that allowed us to access some genomes as well. Now, there's some prevailing issues that we need to take into account, though. So, for example, there is no clear and consistent case definition yet for sugar toxigenic E. coli. Even though it's been with us since the 1980s, it's still very hard for to actually define within the clinical setting. Just because, you know, we have all sorts of different causes of something like bloody diarrhea or kidney failure. So it's actually quite hard to define on its own. There's issues with sensitivity. So for any sort of testing, there always needs to be a selection step. And that then imposes time, and it imposes error, and imposes extra costs and resources. There are a huge number of different O types. And although there is a focus on the most common, that ignores quite a lot of the genetic heterogeneity within the bacteria. So I don't really have time to go into the huge detail just here, but I'm happy to discuss that in more detail if anybody wants. And the current genetic markers are just lack the selectivity that we need. So what we wanted to do was to hold a stakeholder workshop. It was really important to get the stakeholders on board. And then we could actually categorize our genomes. So we wanted to obtain the genomes. We wanted to categorize them in two different bins, if you want, into clinical disease causing versus food that we didn't know if they had the potential to cause disease or not. And then we would scope out different tools to see how we could actually assign a potential burden of disease to these genomes. And we wanted to use a big data or big data capabilities and machine learning tools, for example. and hopefully generate a proof of concept model. And then we wanted to go back to the stakeholders and see what they thought of what we found, and then use that information to outline a larger scale model, bid, sorry. So the first meeting we set up in December, that worked really well. We had a good range of stakeholders, and it was very successful in that we agreed the definitions for what actually was a pathogen and how we would assign a clinical versus potential pathogen. And we also then needed to obtain the genomes as well. So it was really important to get the stakeholders on board to actually give us access to those bacterial genomes. So this is what we got. You really don't need to worry too much about the detail, but what we ended up with was a bunch of non-clinical genomes from red deer survey, from a cheese survey as well, a dairy survey from a cheese outbreak, and also from that retail mint survey. And when we kind of paired those with the equivalent of about 104 genomes that we knew had been responsible for clinical disease. So what that meant is we had around about 200, 230 odd genomes here. But we had, within that 230, we had 130 different O types. So they were really, really divergent in terms of their O classification. And there was very little overlap between the serotypes and the source attribution. So therefore, we couldn't say that all of them that belonged to O type X always came from cheese, for example. There was a huge diversity amongst them. And that's kind of what we wanted to make the query. So then what we did was we carried out two types of genome analysis. And this is what we, the first one on the left-hand side is what we would call an informed type of genome analysis. In other words, we knew what we were looking for. So we were looking for virulence factors, for example, and then we were able to map those to see where they occurred in amongst all of the genomes. So just if you give me a second, I'll talk you through this. I don't know how to make a wee pointer. But if you look on the graph on the left-hand side, we've kind of roughly grouped them into what are the deer isolates, mint, cheese, and the human pathogenic clinical isolates. And wherever you get a spike with the blue and the orange together, then that means that there's a presence of this gene. And wherever you get a gap, that means there's an absence of the gene. So for example, if we look in this green bit in the middle for the mince isolates, there's only a few spikes, so that means only a few of those mince isolates contain that gene, and most of them don't. And in contrast, a lot of the clinical isolates do contain the gene. And that's really reassuring because this particular gene set is associated with a clinical disease outcome. What it tells us straight away is a lot of the deer isolates do contain this virulence factor, so we would actually classify them as probably pathogenic. So that was one type of the analysis. The other type was unguided. So we didn't have any preconceptions like this. We would just simply compare one genome against the other. Now, I really don't have time to go into the detail. The biology, the bioinformatics approach is very nice here. But we have on the left-hand side of this panel on the right, we have a few of the different isolates together, clinical and cheese, for example. There's just a subset here. And wherever they're dark blue, that means the genes are always shared. And then wherever there's white, wherever there's gaps, that means the genes are not present in all of them. They just appear where you get little blue lines. So straight away, you can see how we can use this type of technique to compare those shared genes with the unique genes. And therefore, that might tell us which ones have got a shared set of genes that are associated with clinical disease. Now, the most critical thing in order to do this bit of the analysis is to have that metadata, is to know what your training set is. So what we would say is we already know the clinical isolates cause clinical disease. So anything that looks like that, therefore, has got a strong likelihood it can also cause clinical disease. Okay, and then a third approach is using other machine learning approaches and using bioinformatics tools I don't need to go into the detail here, but we use a tool called PopBunk, which has got a cool name because the bioinformatics tools always tend to. And what was really good about this is we were able to leverage an internship, a 3-month internship from a PhD student, really to query this in much more detail than we could do so in the scoping project. So he was able to look at all of those machine learning approaches, for example. Okay, and then we took all of this back to our stakeholders in the second workshop just recently. So that was held as a hybrid meeting hosted by FTSC in Harwell, and I had a mop-up meeting in Edinburgh as well for those who couldn't attend the hybrid meeting. The same group of stakeholders. So we discussed lots of different things, and one of the crucial things that came up was data accessibility. It was there is a barrier to getting data that is associated with clinical disease, and this is really the same for anybody working on any human pathogen or human disease associated situation. We had also outlined a research project, which was really nice. So, you know, we've got something there which is looking good. We're building that up. And you know, depending on how far we get with the internship, and then hopefully we'll have some strong preliminary data to put into this type of research project. So I'm very pleased with that as well. So yeah, we've done everything we wanted to do. We've generated a really nice collection of isolates that allows us to do this type of analysis. We were able to attribute the source against any potential disease outcome just by using the tools that we had available to us. We were able to identify those common genetic signatures, and the biology looks really interesting there. We were able to leverage this 3-month placement and then outline this research proposal again. And what was really nice about this, and I think where it's worked really well with FTSC, is it's looking at ideas that I never would have really imagined because I just don't have those capabilities. So Martin had some really great suggestions that I wouldn't have known about. So I'm quite excited about that. So yeah, just lots of thanks to the Safe Food Networks, Sonal and Gareth in particular. They've been fantastic, the team, and to all of our stakeholders for sharing their genomes. Right, I'll stop that.
Thanks, Nicola. Thanks for a very insightful presentation. That was really fascinating to know how much you could actually achieve from such a small project. Wonderful results coming out from scoping project. So if there are any questions from audience, feel free to raise your hand, unmute yourself, or even if there are any comments, suggestions, anything is welcome. Yeah, we are pretty much open and flexible in today's webinar. If not, then I just have maybe one a question, and others can chip in as and when they have a question. So Nicola, when you presented the different the data and the genes in different products, I was just wondering, like, can this be also used for some sort of strategic surveillance? Can these data be actually correlated to, let's say, environmental factors of, like, you know, how the products have been experiencing, and, like, you know, where we get more chances of actually like, you know, of these pathogens presence? So what could be those sort of parameters where we could be actually looking for these pathogens presence?
That's a great question, Sonal. So yeah, absolutely. I mean, I myself work really on environmental microbiology, and I'm always looking and thinking about transmission pathways and where these bacteria come from. And this links really nicely to the FSA project PathSafe, where they're really doing a big scale genomic survey of the pathogens, especially E. coli, throughout the UK. And one of the questions there is whether we can attribute particular genomes to particular environments. It's a very hard thing to do for E. coli because E. coli is so successful everywhere. But, you know, there are potentially some hints that we can get from the genomes themselves. So yes, I hope eventually the answer will be yes.
Well, Thank you. But yeah, for the audiences, if there are questions coming up, then please feel free to use the chat box button as we move forward towards the next speaker. And we'll have opportunity as well towards the end to have like an open floor discussion. So you feel free to provide your comments and suggestions there as well. Okay, now we move forward to our next speaker, who is Dr. Maria Anatadaldi, and she's a lecturer in bioinformatics from Cranfield University. She'll be presenting on novel methods for honey quality control. So, Maria, over to you.
Thank you, Sana. Thanks very much. Let me just try to share my screen.
It would be helpful if you speak up a little bit more, Maria.
Okay, yeah, I'll try. I'm in an office, so I'm a little bit mindful, but yeah. So, yeah. And hopefully you can see my screen now.
Yes, we can.
Okay, so thanks very much again for the introduction. Today I'm not going to talk about just one project. I'm going to work about the overall research that I've been doing on honey quality control in the past few years. And I'll try and give you an overview of this. So I have a quite long relationship with the STFC Food Network. I started with a scoping project in 2019, where I looked at a combination of different spectroscopic methods for UK honey authentication. So that scoping project went really well. We had very encouraging results, and this led to a PhD project with Food Biosystems, TTP. And since then, I also got another scoping project and a proof of concept project. So I'm working on a scoping project with the Africa Honey Consortium, where we're trying to find low cost and rapid methods for pesticide detection in honey to address this safety issue. And recently we started a new proof of concept for developing a new testing methodology for honey authentication. And this is also supported by the Food Standards Agency. So honey is a completely natural product, which is a part of the human diet for thousands of years. Despite the prevalence of other sweeteners, both natural and synthetic, in recent times, honey is still a very popular food. Actually, its popularity keeps increasing. So in the UK, we need to import about 40,000 to 50,000 tons of honey per year. And despite the higher price, monofloral honey is actually very popular as well. So the reasons of honey's popularity is the flavor. So customers, the consumers appreciate it for its complex and unique flavor, but also for health reasons. So for the health properties of honey. The ever-increasing demand for honey in combination with the decline of bees inevitably leaves the scene open for fraudulent practices, which means that honey is among the 3 top products in terms of being at risk for adulteration. And at present, there is no single method for authenticity testing, which is a problem that we're trying to address. So what are the challenges around honey authentication? Honey is a very complex matrix. So one is around botanical and geographical identification. So when we buy acacia honey or chestnut honey or heather honey, we sort of rely on the label. And this is what the beekeeper most times thinks that the honey comes from, that the botanical identification of the honey is based on the estimate of the beekeepers. But that's not always an accurate estimate. So this is not because they're trying to deceive us. This is because they're not always sure where the bees have foraged from. But apart from this, there's also the honey fraud problem. So so this is when there is purposeful either mislabeling or in terms of geographical and botanical origin, but also adulterating purposefully the honey with substances that could be sugar syrups, plant-based sugar syrups, or mixing premium honey with cheaper type of honey, or from different geographical origins that it designates on the label. So this is a very widespread practice, unfortunately, especially when it comes to imported honeys. Several tests that have been done over the years have revealed that the majority of imported honey is probably adulterated with sugar syrups. But the issue is that because there is no consensus in the testing methods, these results are often disputed, and very often there is no action taken because there is no precise test that can prove that they have actually been adulterated. And then there's some safety concerns around honey. So the main safety concern is the presence of contaminants, of toxic contaminants in honey, so such as pesticide residue among them, contaminants like heavy metals, for example, or antibiotic residues, which could have been there due to some treatments for the bees used by the beekeepers. Here I have a map for the which shows the widespread honey contamination with neonicotinoids. So this is a class of pesticides which is particularly toxic to bees. So it's not just a concern for human health, but also for the animal health. And despite the ban in Europe and in the UK in recent years, there's still some contamination with neonicotinoids in the honey. Fortunately, most of the levels detected were below the limits set by the EU, so they're below the safety limits. But still, it shows that the presence of pesticides is quite widespread. So it is important to be able to trace and quantify the presence of pesticides. And then another safety concern is the presence of microbiological safety of the honey, and this is mainly because of potential contamination of honey with spores from Clostridium botulinum. So I said earlier that there is no consensus in authentication method for honey. In fact, there are myriads of tests that you can do, and still you might still not have a definite answer. So there are authentication tests that can show you botanical and geographical origin, and then there are tests that can show you whether the honey is, or indicate whether the honey has been adulterated. So this can broadly be divided into classical methods, which is melisopalinology, by looking at the pollen grains and deciding the botanical and the geographical origin of the honey, or doing lab-based methods for physicochemical parameters. Recently, there is a lot of research going on into the development of modern methods using modern instrumentation and methodology. The most prevalent among them is mass spectrometry. The method using isotopic ratio is especially applied for finding adulteration of honey with sugars. There are also spectroscopic techniques such as NMR, FTIR, Raman. And there's also some techniques based on DNA molecular markers. And then when it comes to pesticide detection, the most established methods at the moment is mass spectrometry. So depending on the nature of the pesticide, liquid chromatography mass spectrometry or gas chromatography mass spectrometry. So these are very selective and very sensitive methods. They can go down to very, very low limits of detection. But the problem with both these techniques and the previous techniques that I was talking about, the classical methods for authentication, is the cost and the complexity. So if I talk about pesticide detection, most beekeepers or countries in the developing world do not have access to these techniques and these laboratories. So we need to find some alternative methods which are cheaper, faster, and they can be used in field. And this is what I've been working in the past few years. So I'm developing methods both for botanical authentication and adulteration detection using the combination of sensors with machine learning techniques and also molecular markers. And the other strand of my research is doing pesticide detection using Raman spectroscopy and noble metal nanoparticles. So I would like to acknowledge my partners in this project. So from Cranfield University, I work with Dr. Zoltan Kivey and Dr. Zhujian Yang. And my STFC partners is Professor Pavel Matusek from the Rutherford Appleton Laboratory, and Sarah Mosca, and Dr. Martin Barons from Warwick University. And like I said, we also work with the Africa Honey Consortium. So what is the novelty of these techniques? Raman spectroscopy is suitable for chemical identification. And what it basically gives you is a chemical fingerprint of the matrix that you're measuring. So this can be matched to known chemicals using spectral libraries. The novelty of spatially offset Raman spectroscopy, or SORS for short, is that we can use it to screen substances through containers. So we don't even have to take the honey out of the jar. We can flash the beam through the glass container, and we can take the Raman spectrum. So it's very good for use in field and at all stages of the supply chain for honey. So this is an example of some raw data that we got from different types of honey. And we need to apply some algorithms and some data pre-treatment so that we have nice signals that can give us the fingerprint of the honey matrix. So this is how they look after the pre-processing stage. So when this is done, then we can take the data and apply machine learning and other methods in order to identify the honey. The other method that I'm using for the honey authentication is DNA markers. So this can be used either independently of the source method or in conjunction. So it can provide an additional level of certainty for the results. So what we're trying to do here is we're trying to find DNA barcodes that will be unique for a particular plant species. So this will enable us to do the botanical characterization. And also we're trying to develop DNA markers for plants that are routinely used for adulteration. So corn syrup, for example, rice syrup, sugar beet syrup, these are common adulterants that can be found in honey. So we're trying to create novel and unique barcodes which we can use to say whether the honey has been adulterated or not. So just to give you an overview of the work we've done so far. So when we did the first scoping project, we focused mainly on UK honeys, and particularly we want to characterize the monofloral honeys in the UK. And we focused on heather and borage, which are the most important monofloral honeys in the UK. So we tried to source these honeys from around the UK. We took the source spectrum, and after we cleaned up the data, we applied multivariate analysis, so both unsupervised, like PCA analysis, for example, and supervised, where we use machine learning algorithms to create prediction models. So when it came to botanical characterization, we were able to see 100% separation between the different types of honey. So we had the borage honey, the heather, and also some other honeys, which we categorized as multifloral because the producers could not certify that they came from a predominantly single floral origin. So we were also keen to see whether the source method and the machine learning algorithms were suitable for adulteration detection as well. And we did a small experiment where we could see that by spiking the pure honeys with sugar syrups, we could see the separation, and the method seemed like it could also detect adulteration. So I just wanted to show you a few of our preliminary findings so far. Sorry if I go back and forth between botanical characterization and adulteration detection. We do this hand in hand, and we're actually trying to find tests that will simultaneously detect both the adulteration and the botanical origin. So as I mentioned, we are designing DNA markers that can do this job, basically. And so far, we're working mainly with heather honey, which is the main monofloral honey produced in the UK. And we have been able to find molecular markers that can be specific to the two main types of heather honey. So we have bell heather honey and link heather honey. And we designed 3 types of DNA markers, which shows great specificity for the 2 very related plants. So when we apply the 2 markers in link and bell heather, we're able to amplify the relevant markers for one species, while the other, this marker doesn't amplify for the other species. So we are quite confident that this is the species that we have. And these are some examples from applying these markers to honey. So we applied them to bell honey, link honey, and also mix or honeys that are not heather. And the results are consistent with what we expected to see. So the amplification was consistent with the botanical origin of the honey. So these are very encouraging results so far. And the other strand of our research in terms of safety, we're developing, again, we're using Raman spectrometry again for pesticide detection, but we're applying a specific type of Raman spectroscopy. So the Raman signals are notoriously quite weak. So we can only see the main constituents of a matrix. But in the case of pesticides, we're talking about very small traces of these pesticides in honey. So we need to find a way to amplify the signal that would come from the pesticide. And we're able to do that using surface-enhanced Raman spectroscopy, which uses noble metal nanostructures in order to amplify the signal of individual molecules. So this provides us with the ability to see the signal of these really tiny compounds. And we're trying to...
Maria, just one more minute, and if you can wrap up.
Yes, yeah, sure.
Sorry.
No, I'm about to finish. So yes, we're using this method, and this is the setup that we have for the SARS, and you can see that it's a very small device. So it's particularly useful for using in field. And these are our results that we got for one of the neonicotinoid pesticides, dimethoxam, which is very particularly used in Africa. And here you can see that we have a particular signal that we can attribute to dimethoxam. And the positive thing is that the signal increases with the increased concentration of the pesticide. So that gives us reasons to believe that we can also use it not just for qualitative analysis, but also for quantitative analysis. And we're trying to reach very low limits of detection so that we can sort of compete with the mass spectrometry methods. So in conclusion, our findings so far show that we can get a good differentiation of monofloral UK honeys based on their Raman spectra fingerprint and also their DNA markers. And SOURCE in particular is a promising rapid method for adulteration detection in honey, which in combination with the molecular markers could in the future provide a new testing methodology for honey fraud. And the SARS method again has the potential to develop a rapid and affordable testing method for pesticide detection in honey. Thank you very much for listening, and I'm happy to take any questions.
Thanks, Maria. Quite a lot of data to present from several different projects funded by SFN. So really happy to see so many results coming out in short time frame. I have one question from Nicola. Nicola, would you like to ask yourself, or would you like me to read it?
Yeah, that's no problem. Thanks, Maria. That was really interesting. I was interested to see that you were using the DNA-based detection as well. And yeah, just out of curiosity, what sort of DNA concentrations have you got? I would have thought that it would be pretty low, and you know.
Yeah, so you mean in honey, right?
Yeah.
Yeah, the concentrations are quite low, but we can still, because we're using qPCR, which is extremely sensitive, we can see very good amplification. So even when we have the bands in the gel and they're very faint, sometimes I just squint to see that it's there. Then when we take it to the qPCR, we see very good amplification. So yeah, even when we have and especially when it comes to adulteration, because we would like to be able to find even when it's at low concentration. Then we see quite good amplification still. So yeah, we're quite optimistic.
And can I ask one other question related to that, Sonal? Thank you.
Sure.
And then do you have, a big database then for the genetic markers for the different plant species, so you can kind of screen through them, or, how would you know if it was adulterated by a another non-plant sugar, for example?
Yeah, so with this type of barcoding, we're sort of targeting species. So we're not doing meta-barcoding at this level, where we're not screening for every single species that might be present. But we're targeting the most prevalent types of plants that could be used for honey adulteration. So this is mainly corn syrup, rice syrup, sugar beet, sugar cane. So we're developing methods that are specific to these plant species. we can extend this to include quite wide variety of plants. Also in the future, we are considering using metabarcoding methods so that we can screen for a variety of plants.
Thanks very much.
Thank you.
Brilliant. Thanks, Maria. Well, in interest of time, I have to move on to the next speaker. So if there are any comments, suggestions, anything that you might have also found out from the different projects or the projects that you have been working on, please feel free to use chat box. But we will have opportunities to take up a few questions towards the end. Okay, so our next speaker, in fact, the final speaker of this whole session of webinar is Dr. Manoj Menon, and he's a lecturer in soil environmental science at University of Sheffield. So now we move from honey to rice. So he'll be presenting arsenic in rice. Manoj, over to you.
Thanks, Sonal. Thanks, STFC team, for inviting me to this wonderful webinar opportunity. So I'm going to speak about arsenic, which is a worldwide problem. We have been working on arsenic since 2008, almost using 2 grants. One was from STFC Scoping Award, and the second one simultaneously almost we got a GCRF networking grant. So we have a lot of partners from India and Bangladesh. So we put we call ourselves Arsenic in Rice Research Network, or ARNET team. And we have received 7 or 8 grants in the last few years. I'm not going to show all of those results, a lot of results we have, but I want to focus on what we have done with STFC, and many thanks to STFC's encouragement throughout to develop my research idea. So, I'm from the University of Sheffield, by the way, in Department of Geography. So I'm going to speak a bit about why this is an issue. So arsenic is you can see the atomic number 33, is a very common element in the nature. It's a 20th abundant element in Earth's crust and 14th in seawater, and also even in small quantities we have it in our body as well. But it becomes a problem in some cases. For example, this is inorganic arsenic, which is a carcinogen as well as famous person. He's a king of poisons, they call. Romans called it. And WHO declared this as in the top 10 chemicals affecting public health. And many mystery murders are linked to arsenic poisoning, which is a small pinch of arsenic is enough to kill a person within an hour. So it's very deadly poison. And if you take it depending on the dose intake, you can have different effects on your body. It can be acute effects or chronic effects or long-term effects. So acute effects would be stomach disturbances, maybe you end up in hospitals, and muscle cramping and those kind of things. In extreme cases, death as well. Long-term condition is the one which we normally, especially through food, is the worrying thing, because this is what is happening in Bangladesh and eastern part of India. People are slowly exposed to slowly but long-term exposure is the main issue there through water and rice. So those situations, the symptoms eventually develop into small, like skin cancers, hyperkeratosis is called. So in together they call arsenicosis. All the symptoms linked to arsenic is called arsenicosis. So it's a leading cancer-causing agent of lung and kidney cancers. And it affects almost every organ. If you Google arsenic symptoms, you have a long list of things on Wikipedia, for instance. Particularly infants and children are more vulnerable, and even it will affect its brain. So the main sources are mainly coming through food and water. So this is what, in our project, we focused on food aspect. So if you look at the map of arsenic pollution, is widespread across the world. And you can see little circles on the map. You can see that depending on the density of population, the population affected, you can see specifically around this area, Bangladesh, eastern part of India, West Bengal, state of India, they have, we can call it as, call these places are hotspots of arsenic pollution. But it's across the world, it's it's well spread and well-known problem. Is it's like a pandemic, I could say. It's almost 500 million is estimated to be affected by 108 countries. They are exposed to more than 10 ppb parts per billion arsenic in water. That 10 ppb is the WHO limit stipulated by, for limit in drinking water. So when this all happened, evolved in Bangladesh, WHO made a statement that is the largest mass poisoning of a population in history. Is just because something got really wrong by the tube well program they implemented. So back in 70s, Bangladesh was struggling with children's diseases like diarrhea, and because people were relying on public water bodies. So the UN and World Bank stepped into and promoted tube wells, which you can see one of those in the picture on the right. And this farmer is pumping water out of it. And literally this village we have been, you can see every 10, 20 meters one of those. And in front of every house in their backyard, it's very common thing. And even the paddy fields you can see here, you can see lots of them, not in this picture, but it's a close-up one, but you can see everywhere these tube wells. So when this was promoted, it was with the good intention that this was introduced to provide safe drinking water, groundwater, and actually they didn't test for arsenic. That was a main problem because probably they tested all the common pathogens and stuff in water, but they didn't test for arsenic. And after a decade or so, after this program, people start getting arsenic-positive symptoms. This is the typical skin pigmentation problems you get in this man's hand. You can see palms, thickened symptoms called hyperkeratosis. And this is due to repeated exposure of arsenic through water and food. So before we jump into the technical aspects or to understand my results, so arsenic, we have two forms. One is inorganic form. Chemistry, this is arsenic 3, and they have two oxidation states or valence, arsenic 3 and arsenic 5. This is the one we are worried about. This is the toxic form. And the predominant form is in the water. This is the main thing people just drink, is poison. You can get that also in food, inorganic arsenic. Example, rice is known to take, I'll come to that, but because of this, this is the toxic form, we have regulations for this inorganic arsenic. So for water, as I mentioned, this is the WHO recommendation ppb, 10.1 milligram per liter. And for rice, we have also EU regulations, and this is the same kind of guideline followed in the US, also in China, Australia, New Zealand. Many countries have regulated this now, rice. So rice, for a white rice, it is 0.2 milligram per kilogram, and brown and parboiled rice is slightly more, 0.25. It's still very tiny if you think about it. But for children, the rice which is produced for rice used for infant food or infant food products, it should be rice-based infant food products, it should be 0.1 milligram. So they have almost half of adults legal limit. But there are organic forms of which is called MMA and DMA. The toxicity is still being investigated. At the moment, the consensus is that it's less toxic, or we don't know about it very much. Because this is one of the mechanisms how the body detoxify arsenic. When you take inorganic arsenic, the body methylate it and change into organic forms and then excrete it from the body. So in the food chain, this is a very, very simplified food chain scenario. So the inorganic arsenic, which can come from the tube well, which we mentioned. So the tube well water, which is containing inorganic arsenic, which is used for paddy fields, for irrigating. Rice is a thirsty crop. It needs a lot of water, and the constant submergence is needed for better rice crop, irrigated rice. So essentially that arsenic is coming to the field and then it's taken up by rice because it's not rice to blame, because the conditions in which rice is grown is actually making the rice vulnerable to take a lot of arsenic. And then the grains are consumed by humans, then it can directly go directly through grains, or the straw is an excellent cattle feed. it can be fed to animals, and again, through meat and dairy products, it can reach the humans. So this is a very simplified. There are lots of other products. There are lots of pesticides. They contain arsenic and industrial products. So it's not just one, not this, but there are lots of different sources of arsenic as well. But most commonly, this is in food chain. The main source is actually the contaminated water, which is used for irrigation. So our network is actually work on these UNICEF recommendations, and our idea is to keep these points, let's say minimizing arsenic exposure to crops, develop agricultural practices to reduce arsenic contamination, and awareness. So we have done some surveys in West Bengal as well as in Bangladesh, and we published a paper on how people are conscious about this problem. And also, we very much multidisciplinary approach. We try to work with the different NGOs as well as other charities to help us, and also various academic institutions, and also from within the UK and internationally. So that's our approach, because it's a very much, well, you need a different disciplines to solve this problem. It's not just agronomists are responsible for this. So I'm going to just show some of the results we got from STFC specifically. The first thing what we did, because it's regulated in the UK, the first thing we wanted to check, are the rice sold in the market are they complying to these regulations? So we went and collected lots of rice from different supermarkets and some online. So whatever we could find around Sheffield and some online, we collected 3 major rice types: wild rice, which is actually not a rice itself. Technically, it's a different species, but wild rice, you can see very black at the bottom of this picture. This is very healthy gourmet food, completely different species, but it's very hard to find in the supermarket, but you can buy online. And white and brown rice, you can buy them in supermarket, and you can see the white rice is the most dominant type of rice in many supermarkets. Brown rice less available. And we actually focused on comparing organically and the conventionally produced rice. So that was one of the hypotheses we want to test. And then, so we collected a lot of research papers and, a lot of rice types from a different supermarket. And then we looked at inorganic arsenic. So this is just a summary of the finding. So on this on the x-axis, you can see wild rice, white and brown, and you can see inorganic concentration. So these 2 dashed lines actually show the legal limit. So that's one is the point 2 is adult legal limit, and the point 1 is this infants for infants. So as you can see that there is immediately you can see white rice is probably the safest. It just almost, it has the least amount of inorganic arsenic among all these 3. And brown rice and wild rice, they are neck to neck. Actually, they are quite statistically not very similar, different. But so you can see the brown rice and wild rice have a little bit more arsenic than white rice. But they all of these products we studied in this study are actually complying to the UK regulations, which is good news. Unfortunately, some of them, I think nearly half of them did not follow. It's not very safe to infants. So that was the main finding from the study, that if you are feeding children with these rice types, you need to be careful. So that was our recommendation from that study. You need to strictly regulate the intake of rice because some of them are found to be higher or not suitable for children. And then, but most of them are, almost all rice types, except 1 or 2, I think, if I remember correctly, they are all safe to eat. Every type was following the regulations, which is really good news. So it's extremely, it's okay to eat rice sold in the UK. Although I have to say that we didn't take every single rice sold in every single retail shop. Especially we only covered mostly major supermarkets. So, but you can see rice is available in many other shops, like ethnic stores and stuff. So it's the main message was that nearly half of them are not safe for infant and children. And what is the main risk factor is the amount of rice we eat. It's not the concentration itself. Then we actually, although brown rice contained a bit more arsenic than white rice, but it's full of nutrients. If you look at the wild rice and the brown rice, you can see we looked at the different nutrients: phosphorus, potassium, magnesium, iron, micronutrients like iron, zinc, and manganese. All of them are higher, much higher than white rice. White rice is on the left. You can see those two are brown or brown or wild rice. You can see wild rice is superb in some cases. So much higher, significantly higher amount of nutrition coming from brown and wild rice, because the bran of this brown rice contains a lot of nutrients. At the same time, it contains also a bit of arsenic in it. So there is a risk evaluation we have to be. So we are now at the moment looking at what are the risk of getting, if you switch rice, for example, from white rice to brown rice, what are the risks involved? So we published a paper, we look at the risk factors. Actually, is switching the rice is not the major risk factor. It's actually the amount of rice you eat is the main risk factor. So if you eat lots and lots of rice, like typical average UK per capita consumption is only 15 grams per day, which is nothing compared to a Bangladeshi person who eats 400 grams or something like that. So it's really huge difference in the consumption rate across the world. Asian countries, population there eat more rice. So switching rice is a critical, is not so critical, but the amount of rice you eat is the main thing. So our conclusion was, even if you eat brown rice, you may be getting, but if you eat brown rice, it's not going to increase your risk, because if you're eating less and in optimal way or minimum, on an average consumption up to 200 grams is quite safe to eat. So we totally recommend, because of the nutritional benefit of this brown rice at the moment, is okay to eat them. If you don't eat 400 grams a day, then you're fine. So that's our conclusion was from this paper we published in Journal of Nutrition, British Journal of Nutrition. So, okay, that was one of the, these studies were done on raw rice. So we bought the rice products and we studied them as is. We didn't do any cooking or anything. So we thought one of the ways to reduce the concentration is using different cooking methods, and people have been working on it. So because arsenic is located around the bran on the surface of grain, so, and it's also quite soluble in high temperatures. That means if you cook it with high temperature, it can dissolve some of the arsenic and remove it. So the common, until we published our study, it was cook with excess water was a good method to remove arsenic. So cook with the rice to water ratio like 1:6 or something, then discard that water after cooking. That is a way people do in Asian countries, most of them. So cooking with excess water is a good strategy to remove arsenic. But there is a disadvantage associated. You could lose some of the water-soluble vitamins as minerals as well with it. So, but in the West, we follow absorption method, mostly white rice. So you cook rice with a limited amount of water, allow the water to evaporate, and this is called absorption method. And which because you don't throw any water out, that means you retain arsenic as well as minerals. So it's actually not very good as well. So what we did was, why don't we take the best of both sides and come up with a new method? So what we did was we published a cheeky method that you boil the rice for 10 minutes or 5 minutes or so, because our hypothesis was a short boiling will actually remove that surface bound arsenic from the surface of the grain, and then you discard that water, which is in this third stage. So you boil for 5 minutes, discard that water, and then add just enough to cook the rice further. So that's then from this stage onwards, it becomes absorption method. So which retains most of the nutrients like zinc and stuff. So it was very promising method, and we call it PBA, parboiling with absorption method. So it's a combination of both methods we just discussed. So it retains a lot of nutrients, as well as it removes a fairly good amount of arsenic from the rice. For example, 54% from rice, brown rice, and also 73% of white rice from white rice. So this means this method is quite a good compromise to follow, and it's very easy. It could save some water, and also you cook in low flame, and that means you save some energy. At this point, maybe it's very important. So what we got some award last year, proof of concept award, to continue this work. So we have done raw rice study, we have done cooking study. Now the next stage, we have to go to our gut to see how much of the cooked rice you consume is bioaccessible to you. So this is a work we are in progress. So the title of the project we got now is the potential of brown rice for improving health and investigating the bioaccessibility. We work with the two partners from India, Madras Diabetic Research Foundation. They promote a lot of brown rice products. So we work with them to see how brown rice is perceived among the Persians there, or how the population is actually. It's not popular rice, unfortunately. Even though it's good for you, it's not very popular. And another NGO is going to do some surveys for us as well, summer. They are Chennai-based institutions. So we have started working on this project. We have some results. It's not ready to share yet, so that's why I don't have any results on that. But so far, everything going according to plan. So we're looking at the arsenic and micronutrients coming through our digestive enzymes. So we simulate in vitro digestion to see how much is it bioaccessible. And we are comparing parboiled rice, white rice, and brown rice at the moment, and to see which one is the worst one for bioaccessibility. We look at the risk and benefits, and as well as why the brown rice is not very popular, even though it's a very healthy option based on the data we have. So those are the things we are looking at now. And I think, are we okay with the time?
Yes, I think you are okay to finish. Yeah.
Yeah. Okay, last slide.
Thanks,
Okay,
Manoj.
thanks. Thank you.
That's super helpful, because I'm sure, many of us, we do consume rice, although not the similar amount as we would have consumed if it was a different country. But okay, we have a question from the audience. How Sala, would you like to read your question, or would you like me to read?
Let me drag that question window. Okay.
In the chat.
Yeah.
Okay, I take the mute as an answer that I'll read the question. How would the consumer know there is arsenic on surface of brown rice grain despite the healthy option?
Yeah, this is a trick, because this is a problem. None of the rice is labeled. All we know, they follow the regulations, because consumer wouldn't know any of the nutrient values or arsenic on any of the rice products. And out of these 55 rice, some of them don't even mark the origin properly. I think 23 of them out of 25 did not even say where it came from. So it's also, you see, you don't have the origin, let alone the the nutrient values of it. Yeah, it's not labeled. We recommended this in a paper. You need to have proper label, or you could at least say this is safe for kids at least. So brown rice is in general is good for adults, and it's not very recommended for children at the moment. It's a bit too high for children. Arsenic is a bit too high. So although it's healthy, yeah, it's a healthy rice, and it's actually, I changed to brown rice after I got the results from my own study. So it's really healthy option. Yeah.
But for infants, Manoj, you would still recommend going white rice, or would you recommend using the method that you showed in your previous slides, like, boil the rice and then throw it, and then
I mean, what we demonstrated is, what we demonstrated is that exactly that, because we have been getting, when we wrote the first paper, it was open access. People were communicating direct. Members of public contacted me. I'm having children, what I should do. But that's made me to do work on, okay, this is not something people can rely on. They need to have a method to cook this safely. So that second method is actually quite the one we developed, it became more popular paper, and it is making rice safer. In the paper we described it reduce the risk massively. So any rice, even brown rice, if we cook with that method, or even with excess water or our method, it removes half of the arsenic. That means it's becoming extremely safe to eat, no problem.
Thanks.
Yeah,
Manoj.
for children, I recommend my method or excess water, boil it and throw the water out, whatever the rice is. Yeah.
Thanks, Manoj. That's very helpful. Maria, you have a question?
Yes, thank you. So really interesting talk. It does make us worry a little bit. I was wondering.
Do any of the rice associated bacteria?
No, so we'll read Nicola's question just after Maria. Maria, if you could just speak a little bit.
Sorry.
Yes.
Right.
So can you hear me now?
Yes,
Yeah.
yes, better.
So my question is, apart from differences between white and brown rice, are you aware of differences between cultivars, different rice cultivars in the uptake of arsenic?
Yes, that's we've got a project from STFC, not Food Network actually, STFC 25K. We actually working in Bangladesh on that. So we collected 37 different cultivars grown in a extremely contaminated hotspot, and we haven't published that yet, but we are looking at the cultivar effect. But there is quite a lot of studies looked at cultivar uptake on arsenic. Yeah, there is a genetic difference, but if you look at genetic and environmental factor, genetic has very little control. Even if you put a safe cultivar in a very contaminated spot, you are again risking that. So the environment is a main or dominant factor in deciding this. So genetics, I think if you put a number on it, maybe 20% is genetics, 80% is environment. Those are the two factors actually bringing that arsenic up in rice. Yeah.
Yeah, I say, because my follow-up question would be like if you considered maybe some gene editing, for for breeding cultivars that might
so
less, but if it's such a small factor.
Well, we actually wrote a grant which we haven't got it, but we wrote a grant to BBSRC combining some of these aspects. We wanted to bring in both genetics and environmental factors to look at the screening of cultivars and so forth. And we didn't get it, but it's still something to resubmit.
Well, thanks, Manoj. Nicola, would you like to ask your question?
Yes, thank you very much. That was really interesting. I was thinking, about what you said about transmission of arsenic. So it's mainly waterborne. So, in the field or in the paddies when the rice is growing, we know a lot about the bacteria that associate with the rice plant already. So do you know if there's any sort of bacterial-led detoxification pathways or metabolism pathways that would help naturally reduce, or, are there any magic inoculants that we can add to help reduce arsenic, for example?
I think there's a lot of work going on. I'm not myself working on it, but there is a lot of people looking at the rice microbiome and arsenic. Yes, microbial, everything is microbial run business here. So all these conversions, and even people argue that it could change the toxicity. It could change ion chemistry within the rhizosphere. For example, iron and arsenic on the root surface. For example, I'm going to details. So what we know that the rice has also a lot of can block some of the arsenic coming into the plant. They form a plaque around the root. So this is the iron get deposited to form ferrihydrate around the root. That's why rice roots have this red color. And this is actually a magnet for arsenic. So it's actually, rice is probably doing its best to stop it to going in, but there is also a lot of microbial activities associated with those plaque formation and transformations of arsenic in the rhizosphere. I didn't work on that. I don't have the knowledge very much on this, but I have seen papers on this area. Yeah.
Very interesting.
It's a potential. My collaborator in India actually is a microbiologist. He's a groundwater microbiologist, and he was the one leading the GCRF grant. So he's working on that, for example.
Thank you.
Wow, really fascinating. If there are any other comments or suggestions from the audience, please feel free to unmute yourself and participate in the conversation going on. I have one question maybe for Nicola and Maria. Like when you take samples, it takes some time for getting into analysis and letting us know that there is a pathogen or there is adulteration. Can you give us a rough idea of how much time does it actually take from you taking a sample to you telling us that there is a pathogen or there is an adulteration in the sample?
Sure. Do you want me to go first, Maria?
Yes, please. Yeah, I was going to say.
Okay. Yeah, for the clinical situation, so if it's, a patient who is sick with disease for a verified account of STEC infection, it's at least 3 days. So it takes several days, but because the bacteria is naturally present in cattle and in other grazing animals, if there is surveillance of the animals or in the farm or even of the food, that's going to take longer.
Okay.
So several days at least.
3 days. Okay, thank you. And Maria?
Yeah, so with the spectroscopy method, assuming that we have developed the algorithm, we have the model in place, it's a matter of minutes really, because you just take the measurement and you insert that and the model will straight away tell you. If we do the DNA marker method, I guess about 24 hours, it's the same as doing a COVID test. Hopefully it's much faster.
Okay, and how about you, Manoj? How much time did it take you?
Yeah, I was about to step in and add to that problem. Yes, it's I didn't speak anything about the analytical problems. It's extremely expensive. One of the issues why these countries have problem with the regulations or cannot follow this up, because simply they don't have labs to do that. It's even in the UK, we pay around £100 for one sample. And yeah, you can get water sample analyzed within matter of hours, but the rice, you need to grind them and then it goes through ICP-MS, and speciation of arsenic is more expensive. Yeah, you need a couple of days to get the results. And there is also another risk for the things like honey or any food product. Rice, we don't know where this is coming from. We cannot track grains very easily. And I told you that before, that half of the rice we surveyed did not have in origin. Maybe I am assuming that this is somewhere they procure a lot of rice from different farms and mail it and pack it, and so there's a problem of traceability. So we don't know where it is coming from. And also probably if you buy one rice today and maybe buy the same rice after maybe 1 month, maybe you might get a different batch, maybe completely different concentration. But overall, I'm pleased with the results that they're keeping an eye on it. That's good.
Yeah. No, but that's very important point. I mean, so now when I go to buy rice, I'll definitely looking for the country of origin for sure. Because it was a bit surprising for me to know that there is no country of origin. And shouldn't that be like a kind of regulated? There should be a norm to have a country of origin.
Surprisingly, UK is an important exporter of rice, even though they don't produce a grain of rice here. UK bring rice, brown rice from other countries or maybe the paddy, and they mill it and sell it. I think that's how the origin is lost. That's one theory I have, because it's a very important exporter of rice, milled rice. But I think I haven't seen any rice sold here from Bangladesh, for instance, one of these hotspot areas. I think they're all keeping a close eye on where it is produced. And I think import is only from safe areas. It's definitely linked to the geography, geology. So I think it's a bit more strategic in selecting where it is coming from. And there are studies which came out from different groups of people working on arsenic in the UK. They also looked at different countries and continents and things like that. So these national scale studies are going on everywhere. But it's impossible to say where it, even though it's say, let's say country of origin is India, you don't know where it is still. It's a big country.
Because of course, like eastern part of a country would have the different arsenic concentration versus the other.
But I think the either sellers or here we have a strict control on it for sure, because we didn't find any issues with any of the rice, which was quite impressive. I was expecting a bit of surprise, but no.
So do you have a set of recommendations that like for policy making coming from your papers or from your reports that we can actually maybe discuss with some of the policymakers?
We did recommend the first paper we wrote at least some kind of color coding or some kind of traffic light system safe for children or something they can put it on, arsenic safe or something. Don't have to be actual value printed on, because they wouldn't understand, people won't understand. But labeling could be some kind of flagging up on, is it safe or unsafe for children? For example, simply put something information on that. Or even the cooking method, they could say, okay, use this way to cook so that you remove arsenic. But
they would say that only when they detect that there is enough arsenic in that rice. And from your study, you found that possibly it is below the.
They could say that this rice is suitable to maximum consumption rate for children should be this, maybe minimum, maximum 25 grams per day or something. I think it's easily possible. I mean, based on at least the trend we have. White rice, of course, is safe for children, and brown rice they can put a limit. It's good for kids, I assume, but you have to put a limit on how much they can consume. Okay. But I think I'm curious to get some. I'm waiting for my next study, which is the main thing, because all these stages, cooking, removed arsenic, and also finally when it goes to your gut, that's what it matters.
Exactly. Everything is gut.
So I think that will give us maybe good policy brief afterwards.
Oh, definitely. We are looking forward to for that proof of concept study to come out from you guys. Fantastic work there, Manoj and Nicola, Maria. Absolutely, these are very, very good studies, and I'm sure, people from the policy, whether there's FSA, DEFRA, they are keeping an eye on what's going on in SFN and the different projects that are related to some of their research priorities. So we really hope that we see some follow-on studies after this to make sure that we have a larger impact and impact with the which can change something if it needs to. We change. So with this machine learning, Nicola and Maria, that you guys are working with this machine learning or algorithms that you might be developing in future from these studies, do you think the time that it is taking from taking the sample to analyzing the sample, it would be less? And if yes, less, then how much more less?
Well, absolutely. I mean, if what we want to do is to use the machine learning techniques with very large data sets in order to be able to make an informed signature, and then a genetic signature, for example, and then just simply put those genes of interest into a little lateral flow device ultimately. And then you could actually have, half an hour, 15 minutes, 2 hour turnaround time on the field or by the bedside. So that's the ultimate long-term aim. But yes, the machine learning bit is, in order to get that genetic signature.
Absolutely, that's fantastic. Thanks, Nicola, for that. Any further comments, Maria?
Yeah, and Nicola is right in the sense that we definitely need to build the algorithms using large data sets. So that's the key point there. So that's the time-consuming part. but when you have this database, and then it's a matter of minutes really. So that's our goal, is that in the next couple of years we'll be at this stage.
Brilliant, brilliant. Now these are some of the fantastic results coming from very small studies, so short time frame studies. So really congratulations to all the team members that you are leading, and please pass on my congratulations to them. Fantastic work. We are super proud of all the projects that you have presented today. So I would like to thank you all for your time. I would like to thank Eric and Paulina for arranging this webinar, and thanks to all the audiences and participants who stayed here for so long. Thank you, and we hope to catch up soon. We hope that the conversation sorry, Maria.
I just want to say thank you for the support.
No, yeah, most welcome. No, I really hope that the conversation which we have started today doesn't end here, and we build on to these conversations. We really like to see the synergies coming from the different projects together, and see if we could work forward towards, like, the bigger and the larger grants, which can have some sort of solid impact as well. Yeah. So well done, everybody. And looking forward to engaging with you all sometime soon. Thank you
very
for
much.
your time, guys. And thank you. Bye.